speaker, but we can bring you back up towards the end of the session to get to the rest of those questions.
Happy to do that.
Great. All right. Thank you, Ajay. You just turn your camera mic off. Perfect. Next up, we have Dr. Jayne Aiken. She is a postdoctoral scientist in the Holzbaur Lab in the Probman School of Medicine at the University of Pennsylvania.
Hello, everyone. I'd like to thank the organizers of this exciting KIP1A engagement conference for inviting me to give this talk. My name is Jayne Aiken, and I am a postdoc in the Holzbaur Lab. And I'm excited to share with you today some of the work that we've been doing to help uncover KIP1A's cellular function. To start, I'd like to zoom out on the incredibly complex nervous system, which is formed from billions of specialized cells called neurons that must communicate with one another by sending signals through their connections called synapses. As all of us here know, KIP1A is a kinesin gene expressed in neurons and can become mutated to cause numerous diverse issues with nervous system function. In the Holzbaur Lab, we've set out to discover the important cellular roles of KIP1A and why it's necessary to keep neurons healthy. So let's back up to properly introduce the protein of the hour, KIP1A. What is it and why could it be important? For this audience, I'll keep it brief. KIP1A belongs to a class of proteins called kinesin motor proteins that this video shows in an artistic rendering walking along the microtubule. Which is its canonical job. It walks on the microtubule to disperse cargo. And that's what we think KIP1A is doing in the neuron, carrying neuronal cargos along the microtubule to distribute the cellular components throughout the neuron. We know that without fully functional KIP1A, the nervous system is not able to function properly. So what KIP1A cargos could be indispensable for maintaining healthy neurons? Work from a variety of labs have demonstrated that KIP1A cargos are numerous and diverse, spanning important proteins, organelles, and receptors. Of note to us was the presence of many cargos shown in red, which are destined for the synapse, which, to remind you, are the connections between neurons that enable signals to be sent from one neuron to the other. We've known for a while that KIP1A traffics synaptic cargos, but how does KIP1A know to release those cargos at the synapse? This question is especially important because synaptic connectivity is more complex than what I've displayed in my cartoon so far. The synapse doesn't just form at the end of the axon, but also along the axon shaft, with a single axon able to house thousands of synapses. Pedro Guedes-Diaz, a former postdoc in the Holzbaur Lab, set out to address this question and discovered a novel interaction between presynaptic microtubule regulation and preferential KIP1A detachment. Using fluorescent tags, we can see moving cargos and also track the ends of growing microtubules. Position of these moving dots or microtubule comets can be tracked over time to generate chimegraphs, which shows the position of the thing of interest as it moves over time, which reveals how fast it's going as well as its direction. Pedro was able to use these chimegraphs to analyze the movement of KIP1A's synaptic cargo in rat neurons. And he revealed that these cargos tend to pause and are often retained at these synaptic sites. And interestingly, these presynaptic regions are also enriched for microtubule growing ends, suggesting that the presence of these microtubule ends could be directing the offloading of KIP1A's cargos. My data in human neurons, which are differentiated from induced pluripotent stem cells, also backs up this finding. I find that synaptic cargos frequently stop at presynapses, and that suggests that this mechanism is conserved from rats to humans. More vesicles tend to pause at these synaptic regions compared to non-synaptic regions. And when I look at both synaptic cargos as well as microtubule growth in the same neuron, I find that these presynaptic sites where cargo is being maintained are also enriched for these microtubule ends, which is quantified here. So this prompts the question, how does KIP1A react when it reaches a microtubule end? And to answer this question, Pedro looked at purified kinesins on individual microtubules in a simplified system outside of the cell. And he discovered that a different kinesin protein, KIF5, unlike KIF1A, actually detaches in an unbiased manner all along the microtubule, as shown by these blue top dots, which represent an individual detachment event. KIP1A, conversely, actually tends to preferably detach at the end of microtubule, shown by the majority of these red dots found at the end of the microtubule. So KIP1A tends to walk along the microtubule until it reaches the end, where it preferentially detaches. Pedro further identified a KIP1A disease-associated mutant, T258M, that holds on to these microtubule ends instead of detaching, and found that neurons expressing this mutant have weakened synapses, shown here by decreased presynaptic release. This suggests that appropriate unloading of KIP1A at microtubule ends in presynaptic regions is important for synaptic function. So this story reveals an exciting relationship between presynaptic microtubule regulation and KIP1A mediated cargo delivery. But beyond the synapse, we are interested in uncovering which KIP1A functions or cargoes is really the linchpin for continued neuron health. To this end, we've developed a system that will allow us to probe the cellular function of human neurons. Using the I3 neuron induction strategy in either bulk cultures or in microfluidic devices, which are shown here, that helps isolate axons from dendrites, we can monitor things like KIP1A movement, as well as KIP1A cargo movement in cells to address cellular viability and a variety of other cellular behaviors. And we can do this in neurons that are either wild type, have been depleted for KIP1A. This will give us a baseline for how cells react when there's not enough KIP1A there to perform its job, or in cells that are expressing different classes of KIP1A disease-associated mutants. Molecular data from Pedro and others has helped give us an idea of how these distinct mutations can disrupt function. Some of them are predicted to cause loss of function phenotypes, where it's either unable to bind to cargos or microtubules, or it's not able to move. It could also lead to potentially gain of function issues, where the motor becomes hyperactive or potentially binds to the microtubule but then doesn't move. And expressing these mutants in the human-induced neuron system will provide insight into why the mutants cause cellular defects and ultimately system-level dysfunction. Further, it will help guide therapeutic strategies for distinct classes of mutants. And we can use this model to test whether a gene therapy approach, where you add back in more wild type, can potentially be beneficial for mutants that are predicted loss of function. So with that, I would love to thank my lab, in particular Erica and Pedro, who did a ton of the data that I showed you today, as well as my funding sources, with a particular shout out to KIP1A.org, who recently awarded me a mini research grant to help supply this project. Thank you very much.
Thank you very much, Jayne. Feel free to come up on stage. Awesome. Welcome. Thank you for that great talk. I know I have a question, sort of going back to what we know about KIP1A's interaction with microtubule ends and cargo delivery, like you were reviewing in Pedro's work. What is it about these ends that KIP1A is sensing or picking up on? Is it the actual dynamics? Is it the nucleotide state? And I ask because when we think about non-KIP1A targets for therapeutic development, an idea that comes up often is, well, can we make these microtubule tracks more stable? But based on some of the work that has been shown from the Holzbaur Lab about the really tightened relationship that already exists between microtubule ends and KIP1A, I'm trying to put together in my mind how that may or may not be a therapeutic option if we would be perturbing that.
Absolutely. I love that question because I'm kind of like a microtubule biologist at heart. And so microtubule dynamics are incredibly important for all cells. We think of it, especially for mitosis, you have to have them be dynamic. But we often don't appreciate how the dynamics is important in a more stable situation, such as forming the backbone of these axons or dendrites, which KIP1A walks in both of those environments in the cell. And I think that like just causing microtubules to become more stable in this context might not necessarily be the best thing, because going back to your original question, what we think KIP1A is sensing is in fact the nucleotide state. So while the majority of the lattice is in a GDP state where the nucleotide has lost that third phosphate, at the end, the plus end, it's in that GTP state. And that actually changes the conformation of the tubulin dimers within the lattice. And so we think that that's the case based on some experiments that we've done using different nucleotides. Like we know that it binds more readily to GMP CPP microtubules versus GDP microtubule lattice. I'm currently actually using other nucleotide mimetics like GTP gamma S, which we actually think is probably an even more like physiologically appropriate mimic of the microtubule plus end, as well as different like ion compounds that we can use. So that I think is an interesting direction to go, and potentially not the best idea for like a sledgehammer approach. Let's just stop all microtubule dynamics, hoping that that might help the kinesins, but maybe it'll make them walk potentially better, but they won't deliver their stuff to the right place, which is, you know, is not helpful.
Absolutely. Thank you. One last question. I know we're tight on time, but I wanted to ask sort of a bigger picture question, which is when thinking about certain variants, or you mentioned certain subsets of variants, do you think that different variants are going to have different defects in the cargo that they're transporting compared to other variants? So in other words, do you think we're seeing a scenario where it's like if you have a mutation, your cargo that you transport, it's going to be impaired regardless of what the cargo is? Or do you think that it could be a little bit more nuanced than that based on some of the categories of variants that you described?
So thinking about that, it's interesting for KIP1A because the majority of the mutants are in the motor domain, right? And so when we think of like, oh, different cargoes could be affected, typically that's with proteins that have different cargo domains, and you have mutations in those different domains, and that corresponds to a cargo being left behind and one being not. And so I don't think it's as easy as that to determine with KIP1A at all. And my gut reaction is that for most of these, it's not going to be we're not going to see differences in the subsets of the cargoes. It's going to be either for this mutant, like either all the cargoes are going too far, or these classes, none of them are moving out. That's kind of my gut instinct for this, is like, so potentially for the loss of functions, we might see a buildup of all the cargoes near the soma. And potentially for the hyperactive mutants, you might see a buildup of all the cargoes out towards the axon terminal. Or it's actually interesting, based off of the research roundtable that we got recently from McKinney, he showed that actually with some of the hyperactive ones, they can move without binding the cargo. So maybe you just find a ton of KIP1A out there, but all the cargoes are still left out in the soma. And so then in that case, both the hyperactive and the potential loss of function mutants might lead to very similar cellular defects. But we just don't know that yet. And so by doing these experiments, tracking these different cargoes, we can be able to test, you know, it would be really interesting if, you know, certain mutants had defects in different categories of cargoes. I don't think that's going to be the case, but, you know, by doing these experiments, we'll be able to determine that.
Fantastic. I cannot wait to see how this progresses.
Thank you.
We do have more questions for you, but we're going to kick it over to the next speaker. We'll have you come back up at the end.
Great. Thank you.
Thank you. Okay. And for our